It's my pleasure to introduce you to another of our new next generation leaders, Emily Sylwestrak. She's going to start an assistant professor job at the Institute of Neuroscience at the University of Oregon in spring of 2019, and she tells me that she's going to ride her bike to Eugene for that. So I was impressed by that. Emily's lab will focus on how transcriptionally diverse sets of neurons are organized to mediate complex behavior and how these circuits can be flexibly implemented under different behavioral states. As a graduate student with Anirvan Ghosh at UC San Diego, she studied how cell surface cues allow neurons to recognize synaptic partners and tailor the properties of a given synapse accordingly. As a postdoc in Carl Dieseroth's lab, she developed protocols for visualizing gene expression in intact, transparent tissues in order to merge molecular information with three-dimensional structural information. More recently, Emily has turned her attention to the Habenula, a strikingly heterogeneous brain structure implicated in a wide variety of behaviors, including motivation, anxiety, and sleep. And today, she'll give a talk entitled Linking Molecular Heterogeneity to Functional Organization in Habenular Circuits. Please welcome Emily Sylwestrak.
Thank you. Thank you for that introduction, and also thank you for the invitation to be here today and also to share my work with you. As you'll see, the tools of the Allen Institute have been pretty critical at several points actually in my career, both as a graduate student and now as a postdoc. So this is also just a great opportunity for me to say thank you to all of you for the tools that you've provided. It's an absolute honor to be here, and I'm looking forward to hearing all about the kind of next generation of tools and hopefully to be part of that. The work that I'm going to tell you about today is an unpublished project that I'm finishing up right now in Carl Dieseroth's lab, looking at molecular heterogeneity in the Habenula and how that might indicate a functional organization of the Habenula. But if you'll indulge me for a moment, perhaps we can step back and I'll tell you a little bit about my kind of broader interests and my past work, since I think it kind of dovetails with the interests of the Institute. I've always been fascinated with neuronal diversity. Perhaps this is because I kind of got my start in neuroscience in an electrophysiology lab in which I had to kind of like earn my ability to do any recordings by tracing all of the biocytin-filled neurons for all of the physiologists for a couple years. And that, I think, instilled in me this real interest in the diversity of neuronal morphologies, how vast that is, but then within an individual cell type, you also see this stereotypy. Now that's also reflected in the electrophysiology, a kind of diverse set of electrophysiological properties, but within an individual cell type, there's some stereotypy. And this diversity really enables the nervous system to become specialized. And you see that specialization across several different spatial scales. At the systems level, the brain is organized into functionally distinct units, each processing its own kind of information, and that enables us to kind of deal with the vast amount of incoming information from the environment. But within each of those sections, there's a cellular specialization, where there are individual cell types with defined connectivities, morphologies, physiologies that enable the processing of that information. And of course, there's also a subcellular specialization. Most prominently, the pre- and postsynaptic terminal are incredibly specialized. But what you also see is that individual types of synapses have defined morphologies and physiologies. And that's something I was interested in as a graduate student. And more particularly, one of the phenomena I was interested in was target cell specificity. And this is a phenomenon whereby which an axon, depending on the type of cell it's contacting, so in this case, a hippocampal pyramidal neuron contacting two different interneuron cell types, will build a different kind of synapse. It'll build a facilitating synapse onto one and a depressing synapse onto the other. And because that's the property of the presynaptic terminal, but it depends on the identity of the postsynaptic cell, the thought was there must be some sort of cue, a retrograde cue, that's telling the axon what kind of synapse to build. And although the physiology had been well defined, that molecular cue was not identified. But I was in a lucky position of I was just starting as a graduate student, and it was 2007, and the Allen Brain Atlas existed. And that enabled me to search through all of these in situ expression patterns to identify cell surface molecules that could serve as a cue for this process that were differentially expressed in the two different interneuron cell types. So as a graduate student, that's what I pursued. I identified the cell surface molecule alpha-1, which had been little described, to be selectively expressed in somatostatin-positive OLM cells, and then through further characterization show that that was critical for making this strongly facilitating synapse. So this is an example of how the tools here at the Allen Institute can be really broadly used, and I think in the end, it's difficult to really pin down how many people have benefited from the rich data sources that you have here. I was also fascinated that you just have one gene here that's having a pretty profound effect on neuronal function. It's quite important in this system because depending on these two different interneuron cell types, they target different somatodendritic compartments and route inhibition differently in the hippocampus. So it has a pretty important effect on neuronal function. And in the course of looking through all of these in situ expression patterns in the hippocampus, one of the things that I couldn't help but notice was some nearby interesting transcriptional patterns. And that was in the medial habenula. And it's something I tucked away for a while. I worked on a couple other questions and some technology development in Carl's lab. But I've recently resurrected that interest in the medial habenula because of the really well-described spatial patterning of transcription there. And you can see that here in this set of in situs, the habenula is both a medial and lateral component, and there are a lot of transcriptional differences between the two, but even within the really small medial subdivisions, maybe only a couple hundred microns across, you see a variety of different spatially restricted expression patterns. And just to give you a little bit of an orientation to the inputs and outputs of the habenula, it's an epithalamic structure that is elongated in the anterior-posterior axis and receives a lot of its input from the limbic system, primarily from the medial septum. What's interesting about the medial habenula as opposed to the lateral habenula is it sends its output almost exclusively through the fasciculus retroflexus to the midbrain, to the interpeduncular nucleus, and there it can recruit different dopaminergic and serotonergic nuclei in the median raphe, dorsal raphe, and the VTA. And so I was interested in its ability to kind of integrate the emotionally salient information from the limbic system and then potentially be able to control these midbrain neuromodulatory circuits, as well as this transcriptional diversity. And the thing that struck me about this is it wasn't just that there were, a variety of genes that were differentially expressed, but the kinds of genes that were differentially expressed were ones that you think could have a pretty profound effect on neuronal function. So there are interneuron markers that are differentially expressed, ion channels that might control the intrinsic membrane properties, cell adhesion molecules, including my pet protein alpha-1, as well as a lot of neuropeptides and neuromodulatory receptors that are expressed in different subtypes and have been used to define some of the major subdivisions. There's kind of a major dorsal and ventral subdivision of the medial habenula. So I was interested in this kind of transcriptional heterogeneity that you see here, and I'm going to tell you a little bit about the kind of functional heterogeneity that was also known in the literature. Human fMRI studies had associated the activity in the habenula with painful stimuli, with feedback errors and sensory discrimination tasks, that you see elevated habenula activity in depression, and GWAS studies have also associated with addiction. In the mouse, more recent genetic studies looking in some cases at specific cell types in the habenula, in other cases a total lesion of the medial habenula. You see changes in voluntary wheel running from Eric Turner, as well as primary reinforcement, alterations in lateral habenula activity during depression, and then changes in sensitivity to nicotine, impulsive behavior, compulsive behavior. I also just want to note that the habenula is incredibly well conserved evolutionarily, and you see this same pathway in zebrafish and is looked at in adaptive fear response, social conflict resolution, and anxiety. I tell you all of these things just because there seems to be a lot of different functions ascribed to the habenula. And so since we have this cell type heterogeneity, it's associated with human neuropsychiatric disorders, and there seem to be several distinct behavioral phenotypes in medial habenula lesions. I wanted to know, can we understand something about what the molecular heterogeneity means here, and how that might be giving rise to functional units that are driving these different defined behaviors. So to do that, I'm taking a transgenic approach to look at the activity of different genetically defined cell types during behavior. And these are the 4 cell types I'm going to talk to you about today. Tachykinin-1 are substance P expressing cells, CHAT, TH, and calbindin-1. I chose TAC1 and chat because these are the two best described subpopulations of cells. They're largely non-overlapping dorsal and ventral subpopulations. But when I was looking at all of these in situ patterns in the atlas, what I noticed is that there were several different genes that were expressed more on this lateral edge, kind of the boundary between medial and lateral habenula, including TH and calbinin-1, but also the mu-opioid receptor, cocaine and amphetamine-related peptides, some serotonin receptors. It seemed like it might also be a kind of functionally defined group. To show you the overlap of these, the expression of these cell types that I'm looking at, I've just shown a quadruple in situ hybridization here. Again, you can see the kind of dorsal expression of TAC, in red the ventral expression of chat. If you look at overlap, you can see that those are largely non-overlapping. TAC1 cells express little chat, and chat cells express little TAC. But you have a more overlap in these lateral populations, as well as they overlap with the kind of two existing defined populations. But the goal was to be able to look at these different cell types in behavior. And to do that, I'm using a fiber-based method. So this is using GCamp and an optical fiber just to measure the bulk calcium activity in a genetically defined cell type. So you can see that here. We're monitoring the calcium level as the animal is tethered to the fiber optic. And I'm doing this in an operant training task. So this is one of the a task that had been used in a medial habenular lesion that had shown a change in impulsive behavior, so I wanted to include it. It also has some other nice features in that it has a learned component. It takes a bit of time for animals to learn this task, and there's a reward component, so I thought there were several different things that I could assay simultaneously. Briefly, the task runs like this. Animals have to learn to attend to a panel of cue lights. It's called the 5-choice serial reaction task. And when one of the cue lights comes on, the animal has to nose poke into that cue light and get a reward at the other side of the operant chamber. There's a light above the operant chamber indicating that the animal got the trial correct. Initially, the cue light is quite long, and then it becomes short, and as that happens, the task becomes harder and harder, but animals learn to attend to the cue and then occasionally even make these premature pokes as they're anticipating the cue light coming on. So here's an example of some fiber placements in GCamp expression from the 4 different cell types, and then just a raw trace of an individual trial, and then an example session. So this is 80 trials in a session, z-scored and pseudocolored as shown here. What I really wanted to know were the consistent changes in activity at certain behavioral points in the task. And so to do that, I aligned the fluorescence data to the cue, the nose poke, and the reward. And I'm looking at the activity across these 4 different genotypes, but also sorting by the eventual trial outcome. So if they got it correct, incorrect, if they omitted the trial entirely, or if they made a premature poke. So first I wanted to look at the time period before the cue. Perhaps I could see here if there was some activity that was different between correct and incorrect trials that might indicate a role in attention. I didn't see any strong effects before the time period of the cue. You could see stuff happening a little bit later on if I now realign to the poke, which comes immediately after the cue, and look before the poke, and this might indicate a role in impulsive behavior. I was specifically trying to look and see if there was any difference between correct and premature trials, which have the same motor output, but one is a kind of correctly withheld response. I didn't see large differences before the poke, but what you can see when you align it here is these differences after the poke, and that's because those correspond to the reward period, where I see the largest consistent changes in activity. Of course, reward is incredibly salient for this water-deprived animal. So it's not all too surprising to see a reward response here, but I think what's really interesting is that the temporal dynamics of this reward response differ depending on the cell type. So TH cells seem to ramp up their activity as the animal approaches the reward port. These are all time-locked head entry into the reward port, whereas the TAC1 cells seem to have the sustained response as the animal is consuming the reward. I don't see large changes in the activity of the cholinergic cells during this task. And the calbinin-1 cells show this modest but statistically significant increase also during the reward consumption. I should just note here that you can see the kind of difference in the temporal dynamics if you overlay the trials, and I'm going to focus on TH and TAC1 for the rest of the talk. I also made sure that this wasn't just something about the animal approaching the reward port, that kind of movement into the reward port. So I compared rewarded head entries, which is the same data I showed you up here, but now I'm showing it against unrewarded head entries. So every other time the animal enters the reward port. during the task. And you see you lose that anticipatory response as the animal enters the reward port here. And interestingly, you see a completely different type of response when the animal enters the reward port and there's no reward to be found. And I was interested to see if this was kind of reminiscent of a negative reward prediction error that you might classically associate with the lateral habenula. That is, it's active when the outcome is worse than predicted. And one of the things that I did was change the task slightly. Which you can see here. This is a TAC1 animal with a simultaneously recorded photometry. There's going to be 2 trials. This trial in which the animal performed it correctly and was rewarded, and even on an individual trial, you can kind of see this ramping up of activity as the animal is consuming the reward. And now there's going to be a second trial that the animal gets correct too, but in this case, everything happens except the water reward isn't delivered. And so you can see as the animal goes into the reward port and there's no water, there's this even stronger response to the withholding of an expected reward. So that's just an example of an individual trial. The data, summary data is shown here. Now I'm comparing 70% of trials that are actually rewarded, and then there's some probe trials in which I withhold the water, the light predicting that the water is there, or both of them. And I'm looking and seeing if the activity segregates along these different trial parameters. And what I see is that for the Th3 cells, you only see this anticipatory response in the 2 conditions, the black and the green, in which the light is present, showing the animal that there's going to be a reward. So I think this is Q-modulated activity in the Th cells. And there are 2 different activity profiles in the TAC1 cells. First, you see this rather sharp, sudden, and robust response in the dark blue and black. And those are the 2 trial types in which I withhold a water reward. These now dwarf the more sustained response that I see to the actual delivered reward, which is in the light blue and the green. I was interested, here we see that the blue scenario in which animals have a withheld reward, but they didn't also get that information to expect their reward from the light above the reward port, that seems to attenuate the response. And so I wanted to see if the amplitude of that error response, let's say, correlated with the trial history. And so I've plotted the amplitude of these error responses as a function of what the previous trials were like. So if the previous 2 were withheld, or up to if there were 3 correct trials in a row before this omission, and you see this trend towards an increase in the amplitude of that error response given a positive history of reward. And this is just clustered by each, average for each of the animals. But I wanted to test this a little bit more directly, and what I did was, instead of randomly interleaving these withheld rewards, I clustered them all together into a block paradigm. So this is a little bit more like an extinction paradigm, which there's a baseline period where I'm giving them a water reward every single trial. And then at the beginning of the block, I start to withhold every water reward. And that's at this white dotted line. For the Th cells, you can see that the response before the water reward is rather stable. Again, the light is on this entire time. But for the TAC1 cells, you see this really strong response on that very first withheld reward. But after subsequent withheld rewards or subsequent, let's say, disappointments, the amplitude of that response decreases over the course of the extinction block. Eventually, I pick up the trials, the rewarded trials again. The behavioral performance is also changing across the same time frame. So this is the extinction block, and you can see the percent of trials omitted goes up after they stop getting rewarded for these trials. When I started plotting the data this way, one of the things that I noticed is that there were all of these dynamics that seemed to be happening over the course of the session. These are incredibly well-trained animals who've been doing this task for weeks. So the fact that the actual data wasn't stable over a session was a little bit surprising to me. Let me show you that more explicitly here. So now I'm looking at the average across a session. These are just for correct trials, so this is the nth correct trial. For the Th3 cells that are responding to the light, there's a slight increase, but it's more or less stable over the course of the session. But what you see, the thing that I've been calling a reward response is actually probably a little more nuanced than that. They know this task. They're getting rewarded these first couple trials, but the amplitude of that response is actually much smaller. And that kind of builds up over the course of a session. What's interesting is if I look at this same data plotted in the same way, but for the other types of experiments that I've done where there's randomly interleaved withheld rewards, the amplitude of the given rewards changes. So in the context of only being rewarded 80% of the time, the response in the Tac1 cells doesn't build up as much over the course of the session. And if I bring that down to 50, you can barely see a response, even though they're still performing the task and they're still getting rewarded. So this is something I'm interested in following up on in the future. These kind of trial dynamics exist for both the rewarded trials and unrewarded trials. So now if I take the 20% of trials that were unrewarded, you see that the amplitude is actually larger in the context of usually getting a reward than only getting a reward 50% of the time. And so that's quantified down here at a 50% reward rate. There's actually no difference between early and late trials in the session. So I just want to summarize the data I've shown you and then talk very briefly about what I'm looking at doing moving forward. So I've shown you several different cell types in the Habenula and these pathway that goes from the Habenula to the IPN. Cholinergic cells don't seem to be significantly modulated by reward in this task. I didn't have time to show you the data, but actually this decrease I think is related to the movement in the unrewarded head entries. This is also happening. The TH cells, I believe, are responding to the cues, predicting reward that ramp up activity as the animal approaches the reward port. Calbindin 1 cells are responding modestly to reward, and Tac1 cells are also responding to reward, but potentially more appropriate way to say it is responding to the outcome, because they're responding both to rewarded trials in this more sustained way, but then they also have this higher amplitude transient response to withheld rewards. And interestingly, in the context of occasionally withholding the reward, the response to the rewarded stimuli is decreased. So I'm interested in pursuing the possibility that this activity is modulated by the probability of success or failure in the task. One of the things that I'm struck by is, okay, I'm calling this a cell type, but it's using one gene to define this cell type is probably not sufficient. And I think especially not sufficient because I'm a little bit surprised that one cell would be responding to both a positive and negative valence outcome. And so one of the things that I'm interested to know is if there's heterogeneity within this Tac1 population. There are a couple scattered Tac1 cells in the lateral Habenula, which might be contributing to this withheld response and would be more in line with the literature in the lateral Habenula. And one of the ways that I want to address that is to get single-cell resolution. So I've been working to do grid lens imaging in the Habenula, and in this way I would be able to see, does an individual cell respond to both positive and negative valence outcomes, but also looking at these session-long dynamics, I would be able to see as this activity is building up over the course of the session, is that because individual cells are firing more vigorously, or is it because new cells are coming online later in the session? I'm also looking at getting a better kind of transcriptional resolution in the Habenula. And so I've been applying some of the recently developed in situ sequencing methods in the lab by Zhao Wang and Will Allen. And here's just an example of a small set of sequencing for 16, in situ sequencing for 16 genes in the Habenula. And you can see we do that over 3 different rounds of sequencing, and then can spatial map that, cluster the cells and then map that back to the position. And I do still find a dorsal, ventral, and lateral component here. Interestingly, the dorsal and ventral component are really well defined by Tac1 and chat, which is previously described. And there's a little bit more heterogeneity in the lateral population with these different marker genes that I was interested in. So I'm hopeful that we can get a better idea of what the relevant cell types here and eventually connect that up to the single-cell resolution imaging data to be able to understand exactly what activity motifs correspond to what cell types. And lastly, I just want to acknowledge the people who have contributed to this work. Again, Zhao Wang and Will Allen have been incredibly helpful in applying the in situ sequencing to the medial Habenula. Sam Fasuna is working with me on the grid lens imaging and the great technical support, as well as, of course, Carl's endless support in pursuing these projects. I want to briefly also mention in a project I didn't have time to talk about, the very open collaboration of Harry Choi and Niles Pierce with the HCR method for in situ hybridization. They make all of their tools incredibly well annotated and publicly available before publication. And so I think in the context of open science, they're just great resources to have and great contributors to the community. And I thank you for your attention and take any questions.
Thanks, Emily. Great talk. Questions?
Hi, I noticed with some of the cell types you were measuring right before the input was being given, there was this downticking activity. Is that something cool, or?
Yeah, I haven't looked. So when I collected all of this data, I don't know if you noticed on the animal, I have like this little triangle in hopes, I've also collected video for all of this. And one of the things that I think would be really nice is to get a more comprehensive analysis of that and to see if I can pull out things in the behavior that actually might be well correlated with those kind of more subtle changes in activity, but I haven't done that yet.
Hi. Do you think that for the TAC1 neurons, the fact that every time when you start the session, they are almost at base and then they build up during the session, do you think that they just forget what happened in the previous day and relearn it? Or do you think that there is an history of the fact that you use multiple trial types? And have you looked at what was the type of session which was in the previous day? Because if you've got one with a different type of probability of reward, they might only learn the general task structure and they learn the exact probability of reward for that particular session in that time, and that is the learning you're seeing.
Okay, there's a couple different things. So the first is that the behavior isn't that different on the first couple of trials, so I don't think that they forgot what's happening. When I give these sessions that have occasionally withheld rewards, or especially for the sessions with an extinction block, I always interleave that with days that go back to the typical task that they've learned that they're rewarded 100% of the time. But when I started looking at the modulation with the percent reward, now what I'm interested in doing is paring down to, it takes weeks to train them on this more complex task where they actually really have to attend. But now, since it seems to be a Pavlovian just reward response, I'm going to train them on a much simpler task, and I really want to play around with a lot of those parameters. So for example, is it that they're accumulating this evidence over the course of a session, or is it really about like the intra-trial interval that you get a reward? So if I just paused the experiment for 30 minutes, where would it pick up? Would it pick up at this elevated level, or would it go back down to baseline? I think as soon as I get a little bit of an easier task that I can iterate through a lot of these different possibilities, I'll be able to nail down what is it that they're encoding? Because I wouldn't really say necessarily that's a reward. It's not the appetitive aspects of reward. I think it's maybe more something about the reward probability or reward context of the environment.
It might be interesting to broaden the behavior as opposed to drill down in the neural network, because you started out by talking about all the different behaviors that the Habenula might be involved in, like what if you put nicotine in one of the reward, deliveries, or if you had a model of depression or other things that the Habenula was meant to, you might see these other groups light up like crazy.
Yeah, exactly. I mean, I think there's the question of, for a particular cell type, how much do you want to drive down, right, into what exactly am I doing? And then, but stepping back at the same time and saying, can we get this kind of gestalt view of what the Habenula is doing? And especially for, you mentioned nicotine, I show that there isn't a decrease in activity, but I think I haven't explored that parameter space to see what exactly might be recruiting the cholinergic neurons in this task.
Thank you. I'm not a specialist. You mentioned a function when you were kind of running through the introduction, and it stuck with me. I wondered whether it was known what its relation was to the particular cell responses you're looking at. You mentioned that the Habenula has a capacity to recruit serotonin and dopamine responses. is there a notion of which of your processes recruit one or the other?
Yeah, so that's just by connectivity that I mentioned that. So the interpeduncular nucleus is connected to the median raphe, dorsal raphe, VTA. But the levels to which, if you like stimulate the medial habenula, you might get changes in serotonin or dopamine is not known. I think one of the things that's interesting to think about is that if in the context of having all of these different behaviors, there's one output for the medial habenula, like the IPN. So how is it that you have this kind of divergent behavior? I didn't show you was some of the axonal tracing that I've done in cleared tissue, but this is also obvious in the connectivity data set, in the Allen data set, that depending on the cell type, the axon termination zones and the IPN are very different. So it may be that actually depending on the cell type in the medial habenula, you get a different recruitment of cells in the IPN that might either contact the VTA or median raphe or dorsal raphe. But all of that right now is just connectivity based. I don't know any things that have looked specifically at changes in dopamine or serotonin functionally.
Amazing talk. At one of the last slides, you showed the data of the star map. And I'm curious if you see any subclusters within the TAC1 and TH cells. And if so, are they spatially organized?
So I only have 16 genes here, and they were curated by me, so it would be a bit biased to say that there were subclusters. I think the ideal thing to do would be first to do single-cell RNA-seq to identify clusters, get a curated set, and then apply it to star maps. So I still don't know for sure. With the TAC1, my guess is that it's actually spatially divided, that I'm picking up these with the fibrophotometry, I'm picking up these TAC1 cells in the lateral habenula, and that if I were to have better spatial resolution, I'd be able to see that. But it could be that there's heterogeneity within the dorsal population in the medial habenula as well. I just don't know.
Thanks again, Emily.